Start.
Hello students, welcome to EPG Padasala. I am Dr. K. Gunasegaran from Center of Advanced Studies in Crystallography and Biophysics, University of Madras. Today we are going to talk about the module. The module consists of protein stability, protein engineering. It actually comes from the paper Biocrystallography. In this module, we will cover what is protein structure activity correlation, protein stability, the role of non-covalent interactions in the protein stability, and then how do we do this protein engineering by directed evolution and rational mutagenesis. And we will jump all in a sudden into protein crystallization and then the factors which actually inhibits the protein to crystallize. And then we will discuss about how to prepare crystallizable protein using protein engineering. So when we say this portion, we will say what are all the difficulties in protein crystallization, and then one way we discuss what is protein engineering, how can we do the protein engineering attempts to make a protein crystallizable so that we can do protein crystal structure determination. We all know proteins. Proteins are made up of amino acids, and then we also know one amino acid's amino end will get fused with the carboxyl end of the next amino acid. By the way, it forms a bond called as amide bond, or we call it amide linkage. In proteins we call it as a peptide bond or peptide linkage. In the script you can see the peptide linkage, which is nothing but CONH group, which actually a planar one because of its sp2 hybridization and electron delocalization. We'll go later on. So these amino acids are fused together one by one, which forms a lengthy chain of polypeptide. We call it as polypeptide because peptide units are repeated. And these polypeptides are a lengthy chain, so we don't call still it is a protein because it's just a lengthy chain of polypeptide. It is no meaning, it cannot render any, most of the time it cannot render any function. So it has to be folded to arrive at a three-dimensional structure, then only we call it as protein because now it is functional. Whatever it's supposed to do, it will do. It cannot be performed when it is in the lengthy conformation, so it is folding and then it arrives at three-dimensional structure. Now, each protein is unique, so in its unique folded state, they exhibit function. That's why we call it as now proteins. As we already conclude, protein folding makes a lengthy chain into a three-dimensional structure. And not all polypeptides are going to be folded in the same way. Each polypeptide, based on its sequence, they have a preference and then it's fold in its own unique way. So different types of folding patterns are possible. So each different folding will arise a defined three-dimensional structure. So different folding patterns, so different functional can be exhibited by proteins. So now I am stressing the structure becomes important for its function. So that's why we call it as structure function correlation, or sometimes we even say structure activity relationship. So amino acid sequence which actually determines the structure which actually performs the function. Now we see stability. Now we say it is protein stability. When we say stability, it can be seen in two different ways. One is called as structural regaining capacity. Other one is functional regaining capacity. Means a protein, let's say if it is denatured, and then how quick it can come back to its original state, that is called as thermodynamic stability. And then how quick it retains its function, that's called as kinetic stability. Anyway, in this case we see thermodynamic stability, how the protein gets folded in back to its native form. Now protein folding and then it unfolds. Let's say it is an equilibrium reaction. Protein unfolds and then refolds, let's say. In this case, actually, we know delta G, which is actually Gibbs free energy chain for folding, is G folded minus G unfolded. We all know for any feasible reaction, any feasible event, any spontaneous event, delta G should be negative. So, by assuming this, what we can say, G folded should be lower than the G unfolded. But unfortunately, I warn you, mostly even unfolded protein, it is already in the non-linear one. It is already in low energy. So folded structure should be further lower energy. Then only this can happen. Anyway, so we have to think about this. So the thermodynamic stability is actually determined by delta G folding. And then polypeptide chains is actually because of covalent. Already we said it is because of the fusion of two amino acids. So it's a big covalent connection. But now when we come to a three-dimensional structure, the lengthy chain is folded into three dimension. So now, not covalent interaction, non-covalent interactions becomes very important to bring back it to native conformation. And then when we say non-covalent interaction, there are n number of interactions, right? Hydrogen bonding interactions, ionic interactions, van der Waals interactions, and then so many. Now, hydrogen bond interactions stabilize a structure by reducing an energy approximately 66 kilocalories per mole. Means hydrogen bonding interactions are stabilized in the structure. Now, at the same time, it can, for example, destabilize the structure by 177, that is plus 177 kilocalories per mole. Means any hydrogen bonding interaction stabilizes the energy, whereas any conformational flexibility increases the energy by 177, so it destabilizes the structure. And then one more thing I would like to tell you. In a protein, as I already said, it is a three-dimensional molecule. It is a three-dimensional structure. And then where it lives in the water. Of course, in the solvent, mostly water. So, protein should interact with the water outside, but it should not allow water to get inside. If it get inside, obviously the structure get collapsed. So, always in a protein structure, the core residues will be hydrophobic and then outer waste it is hydrophilic. We all know this. Anyway, in this case, any presence of hydrophobic residues in the core, they stabilize the structure by minus 77 kilocalories per mole. Whereas in the core structure, if there are some polar residues, they destabilize. So, hydrophobic residues inside, hydrophilic residues outside. This is the actual stabilizing mechanism. We already said a protein is folded, and then many non-covalent interactions are stabilizing the interaction. So, the stability of the protein is increased because of so many factors we discussed. Now, for example, hydrophobic interactions, hydrogen bonding interactions and van der Waals interactions, they all reduces the energy of the system by influencing delta H. Whereas conformational flexibility, that is the main factor, because in a structure there are many loops and then they all have freedom, so they can move. That kind of conformational flexibility that mainly increases the energy by influencing the delta S, which is actually entropy change. So actually the folded and non-folded delta G is actually very small one. That small one, that small delta G is because when the non-covalent interaction stabilizes the energy very much, the conformation flexibility try to destabilize the structure always. That is actual contributing factors in the protein folding. And then conformational entropy is mainly rendered by the loopy region. But one thing I want to tell you, unfortunately, loops completely or partially becomes important for a structure by arranging secondary structure elements in their places. Like helices and then strands, they had to come and form a tertiary structure. In addition to that, the same loops partially or completely becomes important for functions also sometimes, because in many active site residues are located at the loopy region. Protein engineering. We said stability, and then we are going to talk about the role of protein engineering in altering the stability of a protein. Normally, proteins are highly biological molecules and then they are very natural. So, stability of a protein is its own intrinsic property, and then it varies, depends on the function of the protein and where it's located. For example, a protein present in the stomach, it has to be with acidic environment most of the time. So, the stability is been determined that way. So, it is an intrinsic property. And protein engineering, what it does, it incorporates changes in a protein molecule to have enhanced activity or even sometimes altered activity by attacking the structure most of the time. And then protein engineering efforts can be adopted to have a stable protein. For example, industrially related proteins. Many proteins are used in the food industries, textile industries and something, and there are many proteins, they are called as therapeutic proteins, which use in the medical world. And then in that case we have to increase the shelf life of a protein, right? And not only that, for example, antigens and antibodies in the case of diagnostic kits developments or even vaccine development. So anyway, these are the area like industry related, therapeutic proteins, antigen and antibodies. We have to use protein engineering to increase the stability of the protein. And protein engineering is not a simple thing, it is a complex. it demands many multidisciplinary efforts, many even biophysical, biochemical for screening efforts, and of course recombinant DNA technology efforts when you're doing the protein engineering. Mostly routine tool used for protein engineering is recombinant DNA technology. Protein engineering step by step. First of all, which protein we want to work on, which protein has to be engineered, that has to be identified. So once the protein has been identified, it has to be purified and then well characterized, what the protein is, what is the size is, and then what it does, what is the rate of the reaction, and then many parameters. So basically we have to characterize the protein completely using biochemical and biophysical tools. Okay, once we did this, now we want to increase the stability of the protein. Then we have to identify what is the way, how to do this. Generally very simple, at the protein level we cannot do any modifications. When we call it as, when we want to modify the protein level, for example, we can add some sugar, we can derivatize it with some chemicals. These are all not protein engineering but chemical derivatization, or even sometimes by changing the environmental factors also we can enhance the stability or even functionality, but these are all temporary. We want a permanent solution, so we go for protein engineering. In the protein engineering it should be done at the DNA level. So now when we say DNA level, obviously it is mutagenesis. So we have to incorporate some mutations to the protein so that it can be enhanced activity or with enhanced stability. This can be done by two ways mainly. One is called a directed evolution and other one is called rational design. And then these are the two methodologies, both are working at the DNA level. But we have to do these two different techniques to perform protein engineering. In the directed evolution, most of the time we do random mutations at the gene level, and then we have to produce the protein during some expression. Then we have to characterize. Then we have to screen whether the protein after this incorporate which particular thing, which particular mutation is giving us the desired function. So in the directed evolution we produce random mutagenesis, then we produce many variants. It's called a library of variants, and then each and every protein has to be screened. In the case of rational design, it is knowledge based. In this case we restrict ourselves to only few mutations, right? Limited number of mutations. From that itself we can screen for the desired protein with the desired functionality as well as stability. When I say select the protein with function or screen for the desired function, that involves many biochemical and biophysical tools. Protein engineering and crystallization. Now, why do we need to crystallize a protein? Because that is the only way to get X-ray diffracted so that we can determine the crystal structure of the protein. So this crystallization becomes very important. Of course, it will bottleneck in biocrystallography. So we have to crystallize a protein, then we can take it to the X-ray diffractometer, then we can get the data, and then you get the structure by doing a lot of mathematical calculations. And when we say what is the role of protein engineering in crystallization, I told you protein engineering is multidisciplinary and then it involves many biophysical tools. I mean, we use which biophysics tool should be used for a particular purpose. Here the purpose is, for example, we want to engineer a protein to crystallize it. Maybe the protein is not getting crystallized, it is wild type. So we need to do some engineering work. So we need to engineer the protein so that it can get crystallized. So that is the purpose. So when we talk about the step by step, so now we have purified the protein, we have characterized it completely. Now we have to do the mutations at the gene level to produce a protein with increased stability. When we say stability here, not with much flexibilities to get crystallized, because crystallization is actually what? It is a kind of gentle precipitation technique. So the molecules are come close together and then they make a cluster and then they organized with some symmetry and then the symmetry is expanded in 3 dimensions. So it's actually a precipitation, but here is a very gentle and then slow precipitation. For that, we need to engineer the protein in such a way, let it come close to one and then let it form a lattice so that a crystal can be grown with that protein. And in a crystal, the packing forces actually which brings the molecule together, right? That's called packing forces. The packing forces by which protein molecules attain rigidness. When we say rigidness, it is no degrees of freedom for the translation and rotation. We cannot make a protein completely rigid, but we can make it not moving here and there. We can reduce it rotation about its axis and then moves here and there. So basically, crystallization is governed by packing forces. These packing forces brings protein molecules to close one another with no degrees of freedom for translation and rotation. This can reduce the energy of the system only by 30 to 100 kilojoules per mole. So packing forces can reduce the energy only to a small extent. Whereas if the protein has a flexible loops or maybe moving domains, then obviously that creates more. The packing forces cannot bring such a molecules together so that it can get crystallized. And one more thing I want to tell you, unlike other biophysical and biochemical experiments, where normally we use a protein, a purified protein, low concentration, mostly like 1 milligram per mole, even less than that. But in crystallization, for crystallization, we need pure, homogeneous and highly concentrated protein sample. Roughly like 5 to 25 milligram per ml. It's a huge quantity, it's a huge concentrated, right? So at that concentration only we had to start the precipitation, then it gives a crystal. So here the another additional problem we have here is we need highly concentrated protein sample for crystallization. Now, in some cases, it has been observed that even in some cases, a stable, a small and then single domain protein, there are many problems in getting good quality crystals. Factors affecting crystallization can be altered by the protein engineering. That's what now we said. So we have to do protein engineering to alter the protein structure. I mean, we incorporate some alterations in the protein so that it gets crystallized. And then as I already said, no, it's a gentle precipitation. So first of all, we have to have a protein with high solubility. Then only we can reach that 5 and 25 milligram per ml. So we have to use a highly concentrated protein. When it is possible? When the protein is highly soluble. So we have to start with the solubility. That is a problem. So the protein should be highly soluble. That protein can be taken for the successful crystallization experiments. In addition to that, as I already said, any flexible loops, if there is any flexible loop, if we feel that is not important for the functionality, maybe we try to delete or alter those regions. So, increasing the solubility and decreasing the disorderness of the flexible loops. These are the two steps mainly we can target in protein engineering to incorporate such alteration in the protein so that the protein gets crystallized. Now we said solubility is an important criteria for a protein for taking it further for the crystallization experiment. Why I said this is low solubility is a big problem. Why? I told you 5 to 25 milligram per ml. That is actual, most of the time we need this such a high concentration for protein crystallization. What will happen if the solubility is less? Most of the time we end up with aggregation and precipitation. When I say aggregation, there are two types. One, sometimes aggregations can be seen, visible aggregates. Sometimes even invisible aggregates can be. That's why we said highly concentrated homogeneous protein for crystallization. So this aggregation is a very serious problem where protein molecules aggregate together, because in crystals molecules are arranged with a periodic manner. That is the actual difference between the amorphous and the crystal. So in crystals, molecules supposed to arrange in a desired orientation, in a symmetrical pattern, but this aggregation will prevent that, obviously. So when the protein has a low solubility, we can always have this problem which is called as aggregation. So we have to overcome this aggregation. And what is aggregation? Molecule-molecule interaction. For example, normally protein is in water, let's say, in a solvent, most of the time is water. So the protein is just moving here and there in water. How? I told you already, in the surface we have hydrophilic residues, and inside in the core region we have hydrophobic residues. So the molecule is happily surrounded by water molecules. Now protein water interaction is more in number. If there are some hydrophobic residues on the surface, what will happen then? Protein interactions becomes dominant than the protein solvent interaction. In that case, protein hydrophobic interaction dominates and then it precipitates. This is called aggregation. So in this case, one of the factors, there are so many factors for the low solubility, but when we say aggregation is an important phenomena which actually reduces the solubility of the protein, then there is a way that we can alter these hydrophobic residues. Means surface exposed hydrophobic residues can be altered, can be changed, can be mutated, so that the hydrophobic interaction between the protein will become low. Now still it can live happily with the water molecule, water solvent. So now aggregation can be prevented by protein engineering. And then one more thing is, one very difficult problem here is how to do this. There are two ways. One is directed evolution, another one is rational design. In the directed evolution, I said it is random mutations, right? Okay, we'll forget about that. In the case of rational design, we need at least informations about the amino acid sequences. So if there is a homologous structure is available, then we can compare with that, then we can select which are the residues. They are hydrophobic residues, they are exposed to solvent. If we pointedly change them into hydrophilic residues, this kind of problem can be solved. And one more thing is, by just looking at the hydrophobicity of amino acids, a protein is made up of amino acids, and then each amino acid has its own hydrophobicity scale. But this individual hydrophobicity scale, using this we cannot compute the solubility of the protein, because I told you already, the protein is a folded pattern or folded form of the lengthy polypeptide. So that's why just sequence information may not be possible to work in this problem. We need a homologous structure is available, then it will be easy. By looking at that, we can select which residue has to be replaced, and then we can go ahead and then we can produce a protein of high soluble. Example, we said solubility is a problem, and then we have to increase the solubility of a protein for better crystallography project. And we also said site-directed mutagenesis can be applied to enhance the stability of the protein by doing some alterations, by mutations. But we said for that homologous structure should be known. In that case we can do. Or otherwise, I already one more also said directed evolution. Random mutations also can be done, and then we can select a highly soluble protein by screening methods. Here there is an example. In the case of ribonuclease, the ribonuclease is from Streptomyces aureofaciens. Here is a beautiful work that has been presented. In this particular protein, there are random mutations has been carried out. But in this case, when I say random, it is like 76th position of the protein is replaced by all possible amino acids. Originally it is 309. Now all the 19 other amino acids has been mutated one by one, and then the solubility alone has been checked for each individual mutants. Now THR76, that is the residue, 30976 have been replaced by aspartic acid, arginine, glutamic acid, serine, lysine, something like every 19 other amino acids. And from the table you can see aspartic acid mutant that gives 43 milligram per ml concentration. That mutant reached up to 43 milligram per ml concentration. Whereas tryptophan, phenylalanine, 309, these are the residues which actually, in the case of 3.6, 4.4 and 5.6 respectively, right? So by replacing THR by 19 different amino acids, extensive work has been carried out, and in individual case the solubility has been measured and then recorded in this table. From this we know how effectively this can be done. The only thing is, it is tedious. Here it is not even directed evolution, it is rational design. We decided to change the amino acid at the position 76, but it is an extensive work. It has been sequentially, serially replaced by one. So 20 different proteins have been purified, and then the solubility has been measured. From this we know the efficacy of protein engineering. By replacing THR into ESP, we achieved the solubility problem. THR originally gives 20 milligram per ml. Wild protein, where the 76th position is THR, the highest solubility is 20 milligram per ml. Upon mutation to ASP, is aspartic acid, which is a polar residue, now the solubility has gone up twofold. It is 43 milligram per ml. So, such a type of mutations can be done to increase the solubility, so that now the protein is eligible to reach such high concentration. Obviously, very easily this protein can be taken for the crystallization experiments. Example 2. In this case, it is HIV-1 integrase, an integrase protein, an enzyme from HIV-1 virus. Here the problem is also same. When this protein wanted to be crystallized, unfortunately, the same problem was encountered, solubility. This protein could not be concentrated more than 4 or 5 milligram per ml. That too, many times it is invisible aggregation, so homogeneous solution could not be achieved for this particular protein. And in this case, almost a directed evolution aspect has been carried out. In the hydrophobic core portion, from 56 to 199, many mutants have been incorporated, and then each and every mutant was screened for its solubility. And then that's the reason here we could see 185th residue, which is originally phenylalanine. From the structure you can see phenylalanine is completely exposed to the solvent area. And then this solvent area happened to be phenylalanine and hydrophobic residue. So this has been screened. This has been mutated with different types of amino acids. Then lysine mutant, means F185K mutant, was found to be highly soluble. They could reach a solubility of 20 or 22 milligram per ml, and then they got the crystal structure from that particular mutant. What I am trying to tell you here is, in the core region 52 to 199, many residues were replaced sequentially, and many different types of attempts have been carried out to produce various mutants. That's why we said it is a library of variants. So many mutants have been generated, and then each and every one had been verified for their solubility, and then they could select this particular mutant, which is F185K, found to be highly soluble, and then fortunately they got the crystals from this particular mutant. Now HIV-1 integrase crystal structure is solved. Why? Because of the protein engineering. What we did? We did sequential mutations. Many mutations have been incorporated, and then from that one mutant has been selected, and then the problem of solubility has been overcome, and then now we have the crystal structure on our desk. We already seen single site mutation is highly helpful for making a protein highly soluble so that it can be crystallized. And there is one more example here. It is human ferritin. Ferritin is a iron storage protein. It has H chain as well as L chain. So we are going to talk about, in this case, is H chain of ferritin, human ferritin. Human ferritin, when they try to crystallize it, it never gets crystals. Mostly it gives only precipitate or aggregates. And then when compared the sequence with the rat and horse ferritin H chain structures, sequences, they found position 86 seems to be very important, because structures were also available at the time. So 86th position happened to be Q in these cases, whereas in human case it is lysine K. So because in the crystal lattice the H ferritin makes multimers, and then it coordinates with some metal ions, and this role of Q is very important in making the crystal contact with the Cu2 plus ions, which actually extends and then makes the crystal. And by understanding the structure of these two sources, that is rat ferritin and horse ferritin, it was decided to incorporate Q at the position of 86 in the case of human protein also. By incorporating this particular mutation, in this case highly knowledge based, and then no random mutations, and then not many residues have been changed. Here directly K was completely replaced into Q, and which actually tremendously drastically changed the crystal pattern, and immediately the lattice was well stabilized, and then they got a very beautiful crystal from which they got the structure of H ferritin also by one single mutation, which is K86Q. It is a monomer of rat ferritin. Is a monomer of rat ferritin. You can see that Q residue getting coordinated with the metal ion. So it's a complete structure. In this case is a macromolecular assembly of L-ferritin formed by CD plus mediated crystal contact. We can see here the monomer of rat ferritin make the multimer. This is what the crystal structure available. From the crystal structure we understood the importance of the residue at the 86th place. By changing this 86th place, which was originally K in human, it has been replaced into Q, then it got crystallized, and then the problem gets completely solved because it gave a beautiful crystal structure by this mutation. This is a beautiful example. Here it is not random attempts have been carried out. Straight away 86th position. We came to know 86th position is important because it mediates the crystal packing via metal binding. So direct replacement of 86 yielded the crystals, and then the problem of crystal structure elimination was solved. So far we discussed about the protein engineering at the gene level, and we had to replace some residues by mutation and all. Now one more thing is, one more big problem with the proteins they resist crystallization is post-translational modifications, particularly glycosylation. Glycosylation is attachment of sugars covalently to the protein. So this chain of sugars may be lengthier one, so that it gives the heterogeneity to the protein sample as well as it gives the flexibility also. By reducing this, one simple thing can be done is you can express the protein in bacterial expression protein, the protein of interest, which is actually glycosylated. When you express this protein in bacterial expression system, the protein will be produced, but unfortunately or fortunately the sugars cannot be attached because there is no such machinery in bacteria. So we can produce unglycosylated version, and then we can go for the crystallization. Otherwise we have to go for eukaryotic expression systems also. And the problem here will be the extracellular. For example, when you do this bacterial expression system, glycosylation will not be there, but sometimes glycosylation may be important for the structure also. We have to be very careful about that. And then there is an example here. The extracellular domain of metabotropic glutamate receptor, it is a neurotransmitter receptor, was subjected to the mutation. Normally ASNX309 or serine. This is a motif for the N-glycosylation motif, right? It is an N-glycosylation site. By mutating this particular motif, obviously we can reduce the possibility of glycosylation. In this case, ASN can be replaced with aspartic acid, so obviously this will not be getting glycosylated. One more thing is, ASN was replaced by GLN in human testis angiotensin converting enzyme to prevent the glycosylation. By that way they prevent the heterogeneity, they got the crystals. And then here we summarize this. Whatever we have learned so far. For a given protein, 3D structure is important to explain the function. To explain the function, we have to crystallize the protein. To crystallize the protein should be highly soluble and all disordered portions should be reduced. Means the protein should be mostly stable. Now we are trying to attempt protein engineering attempts or strategies to make the protein more soluble as well as less dynamic. We are not going to alter its natural dynamicity. We want to remove or we want to alter the flexible portions. Directed evolution and rational modifications. These are the two strategies mainly used in protein engineering. Rational designing works using established knowledge about the structure and function of the molecule. Means that directed evolution is a random, whereas rational design is structure oriented. If we know the structure and mechanism of the protein already, we can directly target such as sites. We can do site directed mutagenesis. We can make the protein altered according to our need. So then we can increase the solubility and then we can make the protein crystallized. So a successful crystallographic projects can be easily carried out by using an important tool or important strategy called as protein engineering. Thank you.